We are very pleased to introduce Dr. Daniel Hayes, the Stuart B. Padnos Professor of Breast Cancer Research at the University of Michigan Rogel Cancer Center and former president of the American Society of Clinical Oncology. Dr. Hayes.
I've been asked today to talk about work we've been doing in my laboratory regarding characterization of circulating tumor cells using the CellSearch assay, going beyond enumeration or just counting cells. I'm fond of and obligated to transparently declare my conflicts. I certainly have conflicts regarding this talk because Menarini Silicon Biosystems has been a big supporter of my laboratory in regards of our use of CellSearch, both laboratory and clinical research funding. Furthermore, the University of Michigan owns a patent, and I'm the named investigator regarding the use of circulating tumor cells, especially phenotyping, and that is licensed MSB, and we receive annual royalties. I have a number of other conflicts which are not related to this talk. Let's start out with some definitions. In the word liquid biopsy is the term is commonly used now principally to refer to ctDNA, but in fact that's not correct. The term liquid biopsy was in fact coined in 2010 by Klaus Pantel and Kathy Alix-Benabriere, who, as defined as research groups are focusing on the clinical value of CTC analysis, and although promising data from patients with advanced disease demonstrate the value of CTC analysis as a liquid biopsy, studies on cancer patients at earlier stages are hampered by low CTC counts. That's all true, and in many respects, Doctors Pantel and Alix-Benabriere were foreseeing the advent of using circulating tumor DNA, but in fact, they were really referring to CTCs when they said liquid biopsy. And I think that's an important issue, that liquid biopsies really refer to anything we could find in blood that might tell us the status of the cancer. Now, I believe that both liquid biopsies and tissue biopsies will be complementary. In terms of liquid biopsies, they're obviously easy to draw. Phlebotomy is relatively inexpensive, and it permits really easy serial testing, whereas tissue, of course, is invasive. You have to really do a biopsy. It's more difficult to obtain. You have to use interventional radiology. It's often very expensive. A pathologist usually needs to be involved, and serial testing, therefore, is logistically quite difficult. The pre-analytical issues of circulating markers are easier to control, fixation, anticoagulation, et cetera, all in a single vacutainer tube, whereas the processing of tissue can be quite complex and relatively easy to screw up, frankly. On the other hand, the sensitivity of liquid biopsies is much lower than what you see in a tissue biopsy. CTCs, and for that matter, plasma tumor or circulating tumor DNA are both really in low abundance, with CTCs being extraordinarily rare events. Whereas, of course, when you do a biopsy, you get millions or even billions of cells in a single needle biopsy. Finally, though, there may be some complementary issues regarding the biology. A liquid biopsy really tells you what's going on in the entire organism, not just the site where you do the biopsy. On the other hand, what's in the blood may not represent the biology of the tissue-based cancer, and vice versa in regards to tissues. So I don't believe any of these is going to replace the other, but rather that they'll be complementary in our regards to taking care of better patients. Now, briefly go over CellSearch. There are really now well over 40 different capture platforms in the English literature, probably more than that. CellSearch is the one that is really at the forefront. It was the first FDA-approved assay for circulating tumor cells and remains really one of the most widely used assays. The pros of CellSearch are it's highly analytically validated, it's highly automated, it's modestly flexible, and the clinical validity, which I'll show in a minute, has been strongly demonstrated. The cons are really less sensitive than desired. It depends on EpCAM capture. You lose CTCs in the process. And again, we're limited in the amount of blood we can draw in a single patient. And so there are probably ways to revise this to make it even better. But right now, CellSearch is one of the better games in town. And this is the closest thing I have to an advertisement. These are just the pieces of equipment and supplies we use to run our assays. Now, most of you probably know how the assay is done. It's based on immunomagnetic particles that are in suspension. and those are bound to anti-EpCAM antibodies. So anything that's making EpCAM gets pulled down by a magnet, and then you come back in with anti-cytokeratin, anti-CD45, and DAPI. DAPI to prove it's a nucleated event, cytokeratin to prove that that event is indeed epithelial, and CD45 to prove that it's not leukocytes. To be clear, CellSearch does not purify CTCs. It enriches them from the billions of red cells and millions of white cells that are in 7 to 10 mils of blood. So then you put it through a laser, and the laser captures each of these things, and it shows you this is a nucleated event. It's an epithelial nucleated event because it's cytokeratin positive. It's negative for CD45, and it puts these together in a composite that a technician needs to go through and say, yes, those are cancer cells, or no, they're not. But importantly, there's a fourth channel. It was originally developed for calibration, but the machine is really highly reproducible, and we found doesn't need calibration. So we can use that fourth channel to do additional phenotyping as long as we can fluoresceinate an antibody, that the antibody maintains its activity with the original protein, and that that protein is membrane bound either within or on the surface of the cell. These are data just regarding enumeration. These are old studies dating back now 17 years when we published our first paper in the New England Journal regarding CTCs in breast cancer, but you can see they are highly prognostic in breast cancer, colorectal cancer, prostate cancer, and then more recently lung cancer. And these have been validated over and over again as a highly prognostic marker. Furthermore, in a study we did in SWOG, what we found is not only is it prognostic at baseline, it's even more prognostic in telling you whether that patient is likely to be benefiting from the therapy he or she just started. These are patients who started first-line chemotherapy in SWOG SO500, run by a colleague of mine at Michigan Medicine, Jeff Smiraj. And the blue line are people who did not have elevated CTCs at first follow-up. These are all metastatic breast cancer patients. The yellow line are patients who had 5 or more CTCs and cleared them within 1 cycle of chemotherapy, meaning they'd had a CTC response. And the gray line are patients who had elevated CTCs and did not clear them below 5 CTCs per 7.5 mils of blood, meaning they did not have a robust CTC response. And you can see that overall survival is clearly different in these 3 groups. Sorry, I meant to do that. So that for arm C, the group of patients who did not appear to have a CTC response, half of those patients were dead within 13 months. 3/4 of those patients died of their cancer within 18 months. And again, this is first-line chemotherapy. So this is a very poor prognostic category. However, in this era of precision medicine, we'd really like to do more than just count cells, but actually look at their biology and see if we can use the markers that we commonly use or new markers to identify targeted therapies that might help us do even better. Let's talk about phenotyping first, and that's where we use the fourth channel. What we found is that we can use that fourth channel for a number of markers, and again, the criteria are just that the antibody has to survive fluoresceination and bind to its antigen, and that antigen has to be membrane bound. It can be internal membrane, nuclear membrane, for example, or it has to be cell surface membrane, as long as it survives fixation. Now, this is a shopping list of a number of proteins that have been reported. The yellow bar are ones that we've done, I'm going to talk more about, and the papers to the right that we've published, principally led by a very, very talented woman in my lab, a medical oncologist who worked in my lab for almost 10 years, Costanza Pelletti, who really pushed this field out to where it is now, in my opinion. The blue is for BCL2 and apoptosis, and again, we've published a couple papers regarding these. And the final one, the green, is PDL1, which we're working on now. We have a paper submitted, but it's not yet accepted or in press. We personally have done IGFR1, androgen receptor, and vimentin, but we haven't published anything on these. Others have. So as an example, the first question we asked was, could we use these phenotyping in the fourth channel to identify women with estrogen receptor positive metastatic breast cancer who will not benefit from the next endocrine therapy? I think most of you know the paradigm for ER positive metastatic breast cancer is to treat patients with serial endocrine therapies, each until it quits working, and then go to the next one. And finally, when a patient appears to be refractory to all endocrine therapy, you move on to chemotherapy. The question is, can we find patients early who are not going to benefit from any endocrine therapy and take them on to chemotherapy where, although it has more side effects, it is probably more likely to be beneficial? So we reckoned that we could use these 4 genes to identify patients who either had sensitive or resistant disease. Estrogen receptor, of course, and also BCL2 probably predict sensitivity to endocrine therapy, whereas HER2 and Ki67 probably predict resistance. And you can see the references below that I've got here that back this up. We then worked very hard to develop an index of reading for each of these markers. And so what we did is we ran a pilot trial of about 50 women. We had to draw a different tube of blood for each of the markers, one for ER, one for BCL2, one for HER2, and one for Ki67. So that's roughly 8 mils of blood times 4, 32 mils. We would pool those and then split them back up into 4 7.5 mil aliquots. You can see here the number of cells per aliquot is pretty close. It's within the variation of the assay. And down below, you can see in this particular patient who originally had ER positive and HER2 negative breast cancer, her CTCs were positive, her BCL2 was positive, her HER2 was negative, and her Ki67 was negative. On the left-hand side, the composite are the actual CTCs, and then that second column is the marker. So that very black background and very white staining implies positivity. And you can see you've got positive ER, BCL2, and positive Ki67 in this case, but lots of gray background and just a smudgy cell really indicates that that was a negative cell for HER2. And from this, we developed a so-called CTC endocrine therapy index, or CTC ETI, with the feeling that the more cells you had, and especially the less likely they were to be ER positive, and the less likely they were to be BCL2 positive, but the more likely they were to be HER2 positive and have elevated Ki67, the more likely they would be to be resistant to endocrine therapy. This particular patient had ER positive HER2 negative breast cancer. She had more cells per tube than the last patient you can see on the top. And what you see here is that in this case, although she had plenty of cells, each of which looks like the first column there, none of these cells was positive for estrogen receptor. So we assigned that patient a very high CTC ETI on the basis that she was ER negative. Her cells were BCL2 positive, HER2 negative, but also elevated for Ki67. So we would predict that this patient might not do well on endocrine therapy. We, as I noted, did almost 50 patients. And Dr. Pelletti worked out this scale on the right so that cells that had no staining in yellow were considered negative. Those that had little staining in pink were considered 1+. Those that had some staining in blue were 2+, and those that were very bright with very dark background were 3+. And what you see here on the top is a series of patients, 11 to 51, it doesn't matter, it's just that patient number, and the number of cells they had per 7.5 mils of blood. And down the bottom, for each of those patients, you can then see the stack of different ER in those cells. What you also see is, except for a few examples where patients were completely ER negative, their CTCs were all ER negative, on the most part, there was great heterogeneity within each patient. I want to point out this patient in particular had almost 1,000 CTCs per 7.5 mils of blood. That's very high. She did not do well. But I also want to point out the enormous heterogeneity that she had so that in her CTCs. So about a third of her cells were ER negative. About another 11% were only 1+. We consider completely negative or 1+ to be negative. And then about half of the cells were 2+, and then 3% were 3+. On the right-hand side, you see an immunohistochemistry of this patient's original primary, where 95% of these patients were very bright ER. So clearly, at least by comparing CTCs when she developed metastasis compared to her original primary cancer, she had developed ER negative clones that were really not very visible in her original primary cancer, consistent with the fact that she was indeed refractory to endocrine therapy. So based on these preliminary data, we started a prospective study of about 120 patients who were starting second or third line endocrine therapy for metastatic breast cancer using this so-called CTC endocrine therapy index. Again, Dr. Pelletti ran this trial, and the results of this trial are now in press. We expect to be published within the next month. Unfortunately, our hypothesis didn't really pan out as well as we thought. On the left, you see outcomes related to baseline CTCs, with the black line being less than 5 CTCs per 7.5 mils of blood, and the red line being greater than or equal to 5 CTCs elevated. And what you see is we expected about a third of these patients had elevated CTCs. That's what we've seen over and over again in ER positive patients. But you can also see that the survival for patients with not elevated CTCs was not quite, but almost twice as great as those who had elevated CTCs, with a statistically significant p-value. What you see on the right-hand side is the same set of patients, but this time broken into CTC ETI. The black curve is identical on the right as it was on the left, because all of the patients who had less than 5 CTCs were considered to have a low CTC endocrine therapy index. But the red line on the left has been broken into those patients who had elevated CTCs, but also had a poor biologic finding in their CTCs, versus those who had elevated CTCs, but didn't have such a bad, basically had a CTC ETI low. And while visually there is some difference between the red line and the green line, statistically there is not. And part of it is because so few patients fell into the low intermediate group. Most of the patients who had elevated CTCs also had a CTC ETI that was high. And you can see that the prognosis in the upper right-hand corner there is worse numerically, but it's not worse statistically for the green and the red. We then we're still interested in that. We're still pursuing it further to see if there are ways we can tweak the assay and make it better. But we've also recently developed genotyping. We and others have begun to be able to not only enrich the CTCs using CellSearch, but then get them to purity by separating them completely from any contaminating white cells using an assay called DEP array. And then taking the cells, amplifying the DNA from each cell, and sequencing them. We hypothesized that a liquid biopsy using circulating tumor cells or circulating plasma tumor DNA would facilitate assessment of somatic mutations above and beyond what you might do with a biopsy. And we have also hypothesized that they could be complementary. And to show you one example here, this is a patient. The columns are individual CTCs. The rows are individual genes. The first two are E-cadherin, and the last three are estrogen receptor, the genes that encode for estrogen receptor and specific mutations. Dark green means a homozygous mutation, both alleles, or one allele deleted and the other mutated. The light green means that one allele was wild type and the other allele was not, was mutated. NC means that we were unable to determine for that particular gene, for those particular CTCs across the board here. And on the far right, you see the tissues. So this patient was felt to have the 2 mutations in CDH1 and to have a Y537S mutation in ESR1. But we also found a mutation that had not been reported before at A569S. In a single cell, but we're pretty sure this was real and not a contaminant, and that was not seen in the patient's tissue. So I've been talking through those. We also found one cell in this data set that was completely wild type, had no mutations at all. We think probably this is a normal epithelial cell that was isolated. Why would there be a normal epithelial cell in the blood? We don't know. We noticed that a long time ago when we first started doing CellSearch. There are occasional wild type circulating epithelial cells that get in the blood for who knows what reason. But it also needs to be kept in mind. And finally, this is a heat map of copy number variations, not mutations. So again, each column in this case is a different gene, and each row is split into patients. So the black line separates each patient, and then each row within the patients is a different CTC. So patient 2, patient 4, so on and so forth. And what you can see just by looking at this is enormous heterogeneity, both between patients, but also for individual genes among the different CTCs. And I want to highlight this particular one. This is ESR1, so of interest to us. And if you just look at patient 2, the top patient, what you can see here is that a couple of her cells had deletions of ESR1, a couple of her cells had amplification of ESR1, and others were wild type for copy number variation within the same patient. This is why we don't cure metastatic breast cancer, because of this sort of heterogeneity. And these are the kinds of things that we're moving forward in now in prospective studies to see if we can begin to identify this kind of heterogeneity and these clones and attack them in a smart way instead of empirically. So I'll end up with this slide. I personally believe that tissue, CTCs, and ctDNA will be complementary for all these reasons, is that you get a lot more cells with tissue, so you could do a lot more genotyping. And you can do candidate assays with CTC. It's a little harder to do with ptDNA. We know that you can phenotype tissue and CTCs. You cannot phenotype ptDNA. Tissue does not represent the whole body. It only represents the area where you stick the needle, whereas liquid biopsies do represent the whole body. We know that tissue represents tissue biology. We've known that for 50 years. The question is, do the circulating markers represent tissue biology and give us insight into patient outcomes and clinical care? And we also know that it's difficult to do serial tissue biopsies. It's easy to do serial liquid biopsies, and that's where we're going with our research. So to conclude, enumeration is highly prognostic at baseline. It's even more prognostic after 3 to 4 weeks of treated. In my opinion, it's a so-called retrospective predictive factor, because it gives you an early insight into how that patient's doing on that therapy. Phenotyping is possible. There's potential is enormous here, but presently it's still investigational. And genotyping is also possible. Again, also presently investigational, but will give us great insight into tumor heterogeneity. So thank you very much. I'm sorry I can't join you in the day that this will be shown. Be happy to answer any questions by email or any other way we can chat. These are the folks who really did the work, especially Dr. Paletti, who has now gone into industry last summer. Emily and Elizabeth, Emily Doltz and Elizabeth Darga are technicians in my lab who did most of the hard work. And then on the right, Scott Tomlin's a great collaborator in our Department of Pathology, and Andy Canny, who did a lot of the genetics and is now a postdoc in my lab. These are the people who supported the work of the BCRF, the Susan Komen, and then AstraZeneca and Menarini. Thank you very much.